genetic diversity and variability of the chestnut blight pathogen in South Tyrol. Please.
So. Where is it? How is it called? I will open. This one? Yeah. Mm-hmm. Okay. Thank you, Renate, for the introduction. Hello, everybody. The topic of my presentation today is genetic diversity and variability of chestnut blight pathogen in South Tyrol. Chestnuts. Chestnut is an important part of European ecosystem. It provides food to the wildlife, it provides timber to us, and not to mention how tasty they are. In South Tyrol, it's an important part of the culture. It promotes tourism in the region by providing these hiking trails, so it attracts tourists. And secondly, it's an important part of the culture. It promotes events like Torglen in the region. But it is threatened by a disease called chestnut blight. This disease is caused by a fungus called Cryphonectria parasitica. This fungus is an ascomycete. It causes canker-like symptoms. It infects cambium and bark tissues. That is why in Italian it's called cancro di castagna. Invasion history of this pathogen. It's an invasive pathogen. The origin of this pathogen is in Asia. It is thought to be somewhere in China or Japan. So any similarity with coronavirus is just accidental. There it does not cause any serious threat to the Asian chestnut because it had the opportunity to co-evolve with the pathogen. Then from there, it introduced to America, and in 1906, it was found in the Central Park in New York. There it killed almost 4 billion trees. That was 99% of their trees, of American chestnuts. And then from there it came to Europe. And in 1938 in Europe it was first found in Genoa, which is a very big port of Italy. And now it is present in all chestnut growing countries in Europe. Then in South Tyrol, in 1958, it was found the first time, and now it is present in all chestnut growing areas of South Tyrol. In 1950s, something else happened. Some scientists observed that the disease was not somehow very strong, the effect was not very strong in Europe. So they thought that it's the resistance. And European chestnuts are more resistant to this disease as compared to American chestnuts because they were not giving up so easily against the disease. But before it was thought that it's resistant, later it was discovered that it was a virus. So this fungus was inhabited by another virus. So this virus causes disease in the fungus. And when this virus is causing disease in the fungus, it loses its ability to cause disease. And that is called hypovirulence, or let's say decrease in the virulence of the fungus. The fungus also loses its color. So normal fungus looks yellowish, orangish, and when the virus is present, it loses its color, and also the sporulation is decreased. And the intensity differs on the basis of which virus is present. So it's an infection. What happens basically that the fungus normally belongs to ascomycete. They have this ability to come together the different strains of the same species. They have this ability to come together and fuse their hyphae and exchange cellular materials. When we noticed that, we started using it as biological control. So it has been happening since past. But this fungus is intelligent. So how it works, basically, this is the graphical representation, how it works. These green dots are viruses. So let's say this is the fungus that is causing the disease in our tree. And what happens that, for example, you have a tree and it's causing a disease in your tree, and you come to me and you say, okay, Farooq, can you please save my tree? And what I do is basically I produce this strain in the lab. It's also Cryphonectria parasitica, but I introduce virus in the lab, and then I go to your tree, and I leave my strain to the disease-causing strain, and what will happen that it will come together, and it will transfer these virus particles to this disease-causing fungus, and it will lose the ability to, or the ability to cause disease will be decreased. But this fungus is intelligent. It has its standard, like how humans have their standards. It says that I cannot just hang out with any Cryphonectria parasitica. I will only hang out with Cryphonectria parasitica that has 6 genes, that has the same alleles on 6 different genetic loci. Okay, so this means that the fusion of hypha will only occur, and the transfer of cellular materials will only occur, and the virus transmission will only occur if all of these genetic loci are the same. This means that there are 6 different genetic loci, and on each genetic loci, there are 2 different allele possibilities. So it means there are 64 different VC types, vegetative compatibility types, or we also call them EU types because they are present in Europe. There are 64 different EU types present. And then this fungus, like other members of Ascomycete, have the ability to do sexual reproduction and asexual reproduction at the same time. So it has 2 different mating types. Think of it as male and female. So the crosses between mate and mate 1 and mate 2 can produce offspring sexually, and mate 1 and will, with the other strain of mate 1, will only produce offsprings asexually. As we know that sexual reproduction is source of genetic variation, so if there is a possibility of sexual reproduction in a region, this means that new VC type combinations will be made in that region. That basically threatens more our chestnuts. That is why it's important to study in order to successfully apply biological control in a region. And it's also important to study the mating type ratios in a region. Then we come to South Tyrol. What do we know? What did we know before this study? We knew that 10 different VC types were present in South Tyrol 25 years ago, and that was the only study done in South Tyrol to study the VC types. We also know that 4 virus-containing strains were applied in this region to control this disease. We also know that import of plants, chestnut plants, continuously happens from other countries, especially from France. And we also know that recently gall wasp was introduced in South Tyrol, and another study showed that abundant galls produced by gall wasp can serve as entry point for this fungus. So basically another opportunity for this fungus to grow. And another study showed that increased mortality rate in South Tyrol. This was because of the 2 reasons that it was a combined effect of chestnut blight pathogen and global warming. Because global warming directly influences the epidemiology of the pathogen, so increased temperature means more possibilities for the fungus to grow. So there are problems in the biocontrol strategies because of the no recent data available. The last data was available from 25 years ago. That is why the aim of our work was to assess the genetic diversity and variability of Cryphonectria parasitica in South Tyrol. So we did extensive sampling all over South Tyrol, wherever the chestnuts are grown. We sampled 36 chestnut stands and 1 forest population. We did isolation of the pathogen Cryphonectria parasitica. We isolated the DNA. Then we did VC type genotyping. Basically, there are 2 methods to study VC types. The one method is for example, I isolate one culture, and I grow it in the lab with all 64 tester strains. That is very lengthy work. The other method is that you just have some markers, some primers, and then you just do PCRs, and then you study which allele is present. For example, this one is allele 1, and this is allele 2. And this is another loci, and it is monomorphic. It is the same in South Tyrol. It is conserved in South Tyrol. So this is another method, and we use this molecular method. The other genotyping we did was mating type genotyping, and we found out that these isolates, for example, are mating type 1, and this isolate, and they are the same isolate here, and this one is mating type 2. So we also used molecular methods here. Then we also did ITS sequencing, because as we know that ITS, internal transcribed spacer region, is very important when you want to study the intraspecific relationships between fungi. So we did PCR purification of the PCR products and Sanger sequencing results. So among 274 different isolates genotyped, we found almost 50% is called EU2. It belonged to one category, one VC type. So that is the most dominant in the region. And the second most dominant, the second and third most dominant was EU1 and EU13. So, for example, now we recommend that if somebody or if the forest department wants to control this disease in the region, we recommend that they use these VC types. They use virus-containing strains of these VC types because they will be the most effective in the region. But then there was another problem, that we also had geographical variation. So let's have a look at geographical variation. And for example, in Wallis Arco, we had a very dominant EU2, of course, it's 73%. But when we started moving to a little bit towards south, in Salto Chiliar district, we had EU2 still dominant, but it started decreasing. And Salto Chiliar was also the most diverse region. 13 different VC types were found here, which is quite fascinating. Then we move further south, and EU2 decreased quite a lot. It is almost equal to EU1. So EU1 and EU2 are equal here, and it co-dominant here in the region. And then in Monticolo forest population, because this was the only forest population, it had the dominant EU1, and EU2 was not very dominant here. It was just 24%. From here, when we start moving again to the north, western north part, again EU type starts increasing, and it's dominant again. And here, if you go further north in Val Venosta, EU2 is quite dominant here. So for example, for better, in order to apply biocontrol more effectively, we can also consider this geographical variation data. And for example, if we want to apply biocontrol in the south or in the Monticolo region, of course, using EU1 will be more effective than EU2. Whereas in the northern populations, Val Venosta or Burgraviato or Wallis Arco, EU2 would be the most effective one. Now let's compare our data with the other European studies. In South Tyrol, found almost 9 on mean value of VC type in South Tyrol was around 9, which is quite high. It's higher than the average Italian. It's higher than almost all other European subpopulations, except Switzerland and Bosnia. It was almost equal to Bosnia, and it was less than Switzerland. So it is quite fascinating that it's, the fungus is quite diverse in the region. Now let's move to the mating type ratios. So mating type ratios tells, for example, if both ratios are 50-50, there is very high chance of sexual reproduction, because it has a very high opportunity to find a mate and reproduce. Whereas if one mating type is dominant, for example, how in Burgraviato only mate 2 was dominant. So for example, mate 2 it will be hard for mate 2 to find mate 1, let's say, and it will not be able to reproduce sexually, so it is better for us. But for example, this data can also be kept in mind when we are applying biological control. So because in order not to introduce another mating type in the region, for example, when we want to apply biocontrol in Burgraviato, it will be better to apply the fungal strains that are made too, in order not to promote sexual reproduction in the region, because that will help for the sustainable application of biocontrol in the region. It will not promote the sexual reproduction. Then the ITS and sequence analysis. So what we did basically that we study our, we sequenced our analysis, and we downloaded all the ITS sequences present in the gene bank, and we did this. We made this haplotype network using the median joining network. It was expected that, of course, this fungus travels 30 kilometers almost per year from one region to another. So it was expected that we would find some relationships between Western Europe or Central Europe because of the geographical proximity. We will probably find some isolates that we found also in Northern Italy or these regions, but it was quite strange that we found. So these orange ones are from South Tyrol, isolates from South Tyrol. And first of all of them were different. So they were different than all rest of the world sequences. Just this haplotype, which was the most dominant haplotype of Cryphonectria parasitica in the world, was also found in South Tyrol, and all these sequences were different. Secondly, it was very interesting to know that these pink ones are from Asia, these pink haplotypes, and South Tyrolean haplotypes were very closely related to these Asian haplotypes. This basically represents that a separate introduction event might have happened directly from Asia. So maybe somebody imported a tree from Asia, and then this fungus might have come with it. Secondly, it was also very interesting to know that this green one represents a European, the other European isolates of ITS sequences. But it was very interesting to know that this haplogroup is basically Eastern European and Hungarian. So South Tyrolean isolates were also very closely related to Hungarian isolates. That was also very unexpected. Another evidence of the relationship between Hungarian isolates and South Tyrolean isolates was found when, as I told you, that EU13 was the third most dominant VC type in South Tyrol. It was also the most dominant VC type in Hungary. And whereas in Switzerland or in Austria, in France, EU2 was the most dominant isolates. So let's conclude the presentation. High VC type diversity of Cryphonectria parasitica represent basically a threat to the local chestnut trees. But anyhow, this study can serve as a basic point to apply biological control more effectively. EU1 and EU2 VC types that were the most dominant VC types shows the relationship between Cryphonectria that was found in South Tyrol and Northern Italy, Switzerland, and Austria. And it basically proves the geographical proximity. Then EU13 dominancy. EU13 was the third most dominant in South Tyrol, shows the relationship between Hungarian population and Asian population. This data basically coincides with the ITS data, and it confirms that Hungarian population and Asian population and South Tyrolean population are somehow linked together, and South Tyrol might have been a bridge between different introduction events in Eastern Europe. A balanced mating type ratio. So mating type ratios differed geographically, but overall in South Tyrol, the mating type ratio was not significantly different from 50%. So yeah, the ratio was 50-50, or 1-to-1 ratio was, the percentage was 50-50, and 1-to-1 ratio was found overall. So this basically represents us that there is huge potential of sexual reproduction. So it also poses a serious threat because in the future, a lot of VC types are expected to be made. The new VC types will be made, will be produced in the region, and because there is no hypovirulent or let's say no virus available in that VC type, they will not have the competition they will have the selection advantage. So yeah, they pose a serious threat to the chestnut also. ITS sequence analysis also confirmed the relationship between Asian and South Tyrolean isolates. And thank you very much for listening, and I would like to thank you to the Department of Forest of the province of Bolzano and Chestnut Growers Association who helped in sampling, and also to Free University of Bolzano who granted the project and the PhD scholarship. Okay, thank you very much.
Thank you, Farooq. Are there any questions about this argument? Sind einige Fragen hier? What about your opinion, the realistic chance to reduce chestnut blight in South Tyrol in the future? How do you think will it evolve?
The realistic chance, of course, is biocontrol, because there is this virus present and it has been working since so many years. There are some challenges, of course, because the fungus is evolving and it's producing new combinations, but of course, by staying up to date, by doing research and by finding what are the exact combinations present, what are the, for example, VC type present in one region, and for example, what are the mating types present in one region, we can basically apply biocontrol very effectively if we know that.
And do you have also a time? Do you know about, is it, are you thinking about years or?
Time?
No,
sorry,
if you apply biocontrol,
yeah.
is it realistic that within years it would be better, or?
The thing is that the biocontrol it's not like the other biocontrol when you apply a different strain and they kill this, for example, this thing will kill it. It's not like that, because this biocontrol is itself the same fungus, okay? So biocontrol is itself Cryphonectria parasitica with virus, okay? So it does not kill the fungus because it's itself, and the virus does not kill it. The fungus will still be here. We cannot finish it. But the tree will survive, the trees will survive, and the trees will not die. And this has been happening since so many years in Europe, and this is how we can decrease the mortality rate, for example. And for that, we don't need a lot of years. When you apply biocontrol, and if it works, it's very quick. It converts into the hypovirulence, and the symptoms, the virulence of the pathogen decreases, and yeah, it is very quick. But if it doesn't work, of course, then the tree can die.
Okay.
Thank you. Sure.
What is actually the method to apply these hypervirulent strains? Is this based on implanting these agar pieces, or have you ever another method?
No, the most commonly used method is just to grow it in the lab and take it to the forest. And because this canker has some cavities inside, so you just take the agar and put it there and then seal the wound with some paraffin.
So you have to reach every canker
in
the wood,
yeah.
on
No.
the tall
For sampling
trees.
also.
So it's very difficult.
For sampling also, we had to climb the trees because these trees are very difficult to collect samples from, and the cankers are very tall. Sometimes I understand, but this is the option we have. But normally, the biggest cankers are kind of located on the stem, so they are kind of closed. But I understand not every canker is here. But also, we don't know, for example, if I introduce the hypovirulent strain in one wound, will it also transfer to the other wound? This is still unknown, but of course, yeah, it is something to be done in the future.
One last question.
I was wondering if you have information about the origin of the virus, Was it already present in the original range of the fungus and was co-introduced when it came to Europe, or did the virus come from somewhere else to create this new relationship with the fungus?
This virus, it is unknown, the origin of this virus, it's not known, but this virus only exists within this fungus. Okay, so it cannot exist outside this fungus. It cannot transmit to humans or other species. That's why we only apply this biocontrol within this these fungal cultures. But yeah, the origin is not known of this virus.
It only exists in Europe, then?
No, it exists in America also. It was tried to use a lot in America also in order to control this disease, but there it did not work because in America there were a lot of vegetative compatibility types. So in America there were 50, 40 compatibility types in a region. So when you have 50, there is no like dominant. For example, here we had like overall in South Tyrol 50% is just EU2. So for example, if I apply EU2 to every chestnut stands here, there is 50% chance that it will work. But in America, there are 50 VC types in same region, so the opportunity is very low. That's why it did not work in America. Welcome.
Thank you, Farooq.
Thank you.
Gehen wir zur nächsten Präsentation über. Jakob Geier von der Versuchsanstalt Leinburg wird uns etwas über die Rolle der Südtiroler Apfelanlagen in der Pollenversorgung der Honigbiene erzählen. Danke.